Hello, class. Welcome to Flourish Nursing Academy. This is Tutor Godwin. Let us look at the anatomy and physiology of the human blood. Blood constitutes approximately 7 to 8% of total body weight in adults, with circulating volumes typically ranging from 4.5 to 6 liters depending on sex, size, and physiological status. Centrifugation of anticoagulated whole blood yields a visible stratification, a lower red fraction, erythrocytes, comprising the hematocrit, a thin intermediate buffy coat containing leukocytes and platelets, and an upper straw-colored plasma. The hematocrit, hemoglobin concentration, and cellular counts are standard clinical indices of blood status. Normal hemoglobin, males, or 13.8, 17, 2 GDL, females, 12.1, 15, 1 GDL. Hematocrit, males, 41 or 53%, females, more 36 or 46%. WBC count, 4,000, 11,000 UL. Platelet count, 150,000, 450,000 UL. Plasma. Composition and functions. Plasma is the liquid matrix in which the formed elements are suspended. It is approximately 90–92% water and contains dissolved solutes, including electrolytes such as sodium, potassium, chloride, and bicarbonate, as well as metabolic substrates like glucose and amino acids, metabolic wastes, gases, hormones, and a variety of plasma proteins. The major plasma proteins are albumin, globulins, and fibrinogen. Albumin, the most abundant, contributes significantly to oncotic pressure, thereby maintaining intravascular volume and facilitating the transport of hydrophobic molecules, including many drugs and hormones. Globulins encompass transport proteins, complement components, and immunoglobulins that participate in humoral immunity. Fibrinogen is a soluble precursor essential to the coagulation cascade. Upon activation, it is converted to fibrin to form a hemostatic clot. Plasma also contains lipoprotein particles for lipid transport, carrier proteins such as transferrin for iron transport, and numerous regulatory factors. When clotting occurs and fibrinogen is removed, the residual fluid is termed serum. Serum analysis is frequently used in clinical chemistry. Formed elements. Erythrocytes. The formed elements of blood consist primarily of erythrocytes, leukocytes, and platelets. Erythrocytes, or red blood cells, are highly specialized cells optimized for gas transport. They are biconcave discs in mammals, a morphology that maximizes surface area for diffusion and confers deformability for passage through narrow capillaries. Mature mammalian erythrocytes lack a nucleus and most organelles, thereby providing ample intracellular space for hemoglobin. Hemoglobin is a tetrameric protein containing heme-prosthetic groups that reversibly bind oxygen. Oxygen loading occurs in the pulmonary capillaries and unloading in the peripheral tissues. Several mechanisms transport carbon dioxide, dissolved in plasma, chemically bound to hemoglobin as carbamino compounds, and predominantly as bicarbonate formed by the hydration of CO2 catalyzed by carbonic anhydrase within erythrocytes. Erythropoiesis, the production of erythrocytes, occurs in the red bone marrow under the regulation of erythropoietin, a glycoprotein largely produced by renal peritubular interstitial cells in response to hypoxia. The average erythrocyte lifespan is approximately 120 days, after which senescent cells are phagocytosed principally by splenic macrophages, and iron and heme components are recycled. Formed elements. Leukocytes. Leukocytes, or white blood cells, are a heterogeneous population of nucleated cells that mediate host defense, surveillance, and inflammatory responses. They are broadly classified into granulocytes and agranulocytes. Granulocytes include neutrophils, the most abundant leukocyte, specialized for rapid response to bacterial invasion. They perform phagocytosis, release antimicrobial granules, and form extracellular traps. Eosinophils, involved in defense against helminths and in modulation of allergic responses. Basophils, rare cells that release histamine and other mediators during certain hypersensitivity reactions. They are related functionally to mast cells. Agranulocytes include lymphocytes, subdivided into B lymphocytes, T lymphocytes, and natural killer cells. B lymphocytes differentiate into plasma cells that secrete immunoglobulins. T lymphocytes mediate cell-mediated immunity and assist or suppress other immune cells. Natural killer cells provide innate cytotoxicity against virus-infected and transformed cells. Monocytes circulate in blood as precursors to tissue macrophages and dendritic cells. They affect phagocytosis, antigen processing, and presentation. Leukopoiesis occurs in the bone marrow and lymphoid tissues and is regulated by colony-stimulating factors and interleukins. Differential leukocyte counts and morphologic examination yield critical diagnostic information regarding infection, inflammation, allergic states, and hematologic malignancies. Formed elements, platelets and hemostasis. Platelets, or thrombocytes, are cytoplasmic fragments derived from megakaryocytes in the bone marrow. They are indispensable for the maintenance of vascular integrity. Primary hemostasis involves platelet adhesion to exposed subendothelial matrix, activation, and aggregation to form a transient platelet plug. Secondary hemostasis is the enzymatic coagulation cascade culminating in the conversion of fibrinogen to fibrin by thrombin, stabilizing the platelet plug into a durable clot. The coagulation cascade has classically been conceptualized as intrinsic and extrinsic pathways that converge upon a common pathway. Calcium ions and vitamin KK-dependent clotting factors are essential cofactors. Hemostatic balance is finely regulated by endogenous anticoagulants such as antithrombin and by fibrinolysis, mediated primarily by plasmin, which degrades fibrin and resolves clots. Blood groups and subdivisions. Blood is further classified by antigenic determinants present on erythrocyte membranes. The ABO system comprises A and B carbohydrate antigens. Individuals express A, B, both, AB, or neither, O. The Rh system, most commonly defined by the D antigen, denotes Rh positive or Rh negative status. These antigenic profiles determine serologic compatibility for transfusion and obstetric considerations. For instance, type O individuals lack A and B antigens and may donate red cells broadly. Type AB individuals possess both antigens and are universal plasma donors. The Rh incompatibility between mother and fetus may precipitate hemolytic disease of the newborn in the absence of prophylactic immunoglobulin. Blood typing and cross-matching are mandatory procedures to prevent alloimmune transfusion reactions. Functions of blood. 1. Transport, delivery of oxygen and nutrients to tissues, and removal of carbon dioxide and metabolic wastes. Blood also